Module 15, the urinary system, acid-base balance in the body. It's now time to discuss the details of how the urinary system controls pH of the blood. You already know that proper blood pH is critical for many body processes. The normal blood pH range is between 7.35 and 7.45. That doesn't leave much wiggle room, does it? And notice it's just above a neutral 7.0 pH, so it's just a bit alkaline. In fact, if the blood pH reaches 7.0, that's considered to be a negative situation called acidosis, which means there's too much acid in the blood. If the pH were to drop anywhere below 7.35, it would cause problems. The function of the nervous system would be negatively affected, eventually causing coma and even death. A person could not even survive if they had a blood pH lower than 7.0, so you can see this factor of the body has to be carefully controlled. Now, the opposite condition of acidosis is when the blood pH is above 7.45. That's a condition known as alkalosis, and again, this situation could cause death. Alkalosis results in the reverse effects of acidosis, causing an overexcited nervous system, resulting in body convulsions. Convulsions occur when skeletal muscles are contracting uncontrollably. If this severe condition continued, the body could not survive because the respiratory muscles would begin to stop regularly working. Now, all of these conditions have to do with the pH of the blood. Remember that cellular pH is a bit more acidic due to the carbon dioxide the cells produce as a result of respiration. Inside working cells, the pH is between 6.0 and 7.4, and that's fine because cells were designed to survive those levels. But the blood has to be within its defined range. Because this is such an important factor of blood, it's a good thing to understand what can cause blood pH to change. One condition that affects it is prolonged gastric vomiting. You see, when you throw up, a lot of the stomach acid is lost. You know there are mechanisms in the digestive system to release basic chemicals into the digestive tract, so they will continue to secrete them, causing more alkaline environments in the gut. That material is absorbed into the bloodstream through the small intestine, resulting in a higher blood pH. Of course, this happens only with prolonged vomiting, not just a few times over the course of a few days. Another condition that can affect blood pH is diarrhea. With diarrhea, the body loses lots of bicarbonate from the intestine, and so severe cases of diarrhea can cause acidosis. Intestinal vomiting is another cause of the loss of bicarbonate. Bile and pancreatic juice are lost, and both of those substances contain bicarbonate. This can lead to acidosis too. You just learned how the kidneys control the amount of hydrogen ions in the blood. Well, if the kidneys aren't working properly, then pH problems can result. So we can say that kidney dysfunction can affect blood pH, causing it to go above 7.45 or below 7.35 based on the types of food eaten. For example, if the diet consists of excessive acidic foods, then the kidneys wouldn't be able to remove the excess acid and acidosis would result. Another problem that can affect blood pH is respiratory dysfunction. Remember in the last module when you learned about the respiratory system, I talked about how this system can control blood pH due to its control of carbon dioxide levels. Well, thankfully, the body has several ways to control blood pH. The first is through buffer systems. A buffer system is a mixture of an acid and a base that resists changes in pH. We've already discussed how buffers work, especially in the digestive tract, to control pH levels. The second way to control pH is through the respiratory system. Again, I already talked about how this is done via carbon dioxide levels. The third way is a slower mechanism, but it's a more complete method of control. This is through the work of the kidneys. You see, kidneys secrete hydrogen ions into the filtrate. More hydrogen ions in the filtrate means less in the blood, and the blood pH rises. When this happens, then, the urine will have a lower pH. Of course, if the kidneys secrete fewer hydrogen ions in the filtrate, there are more in the blood, so the blood pH stays lower and the urine pH rises. Well, of these three methods of pH control, buffer systems is the least effective, but it's still important because it's the fastest response to pH control. You can better understand how buffers work in the following experiment. Your blood pH is controlled by buffers, and the bicarbonate buffer is one of those buffers that helps keep our blood at the correct pH. In this lab, we're going to explore that bicarbonate buffer. Now, the first thing you want to do is you want to gather all your lab materials together, and you want to make sure that your materials are completely clean that you're using. And when I mean clean, I mean washed well with soap and water, and then thoroughly rinsed with tap water, but then you want to do a final rinsing with distilled water. And the reason we need to use distilled water is because we're doing different pHs, if any residue of other material is found in your glasses or in your measuring spoons or even in your eyedropper or pan, you could affect the results of this lab. So I've already washed my hands thoroughly and washed all of our equipment thoroughly with regular water and soap and water, and then a couple of times with my distilled water. And I'm going to finally do that with my pan. I'm going to do a final rinsing here of my pan. And really all I'm doing is adding the water to it and swirling it around to make sure I'm just dissolving any leftover residue, and then dump that in the sink. And then at that point, we're going to start our lab by taking some of this water. We want to put about 3 cups into our pan, and then we're going to add a couple of leaves of red cabbage. Let me place this on the stove. Red cabbage has chemicals called anthocyanins in it that give it this beautiful purple color. You don't need a whole bunch of leaves, just a few. We're going to place them in the pot, and we're going to start this at high and get it boiling. You want to let the leaves boil in the pan for just a few minutes. Once it comes to boil, let it boil for about a minute and a half or so, and then take it off of the burner and let that material cool. So let me get these in there. I've got 2 large leaves. I'm going to put 1 more leaf in there. The rest you can use for a yummy salad. Okay, put those in and get them started. And we will be back after my water has boiled. Well, I've allowed my cabbage to boil for a while, and I've taken it off the burner so it's cooled a little bit. So now what we're going to do is we're going to take the liquid and divide it equally to these 2 juice glasses. I'm going to do that over the sink. And you'll notice that the distilled water has taken on a lovely purple color. If you get some of the lettuce or the cabbage leaves in your glass, that's okay. Just pick that out to remove it. Okay, and they're about the same height. Let me dump some of this one out. This does not have to be completely accurate. All right, so first of all, why is it purple? The leaves contain a chemical called anthocyanin. Anthocyanins are acid-base indicators, and so they will change color whether they're in the presence of an acid or a base. In fact, it turns blue if it's in the presence of a high pH, and kind of a pinkish color if it's in the presence of a low pH. So we were first going to work with one of our containers, and we're going to add a buffer to it. And so the first thing we're going to do is we're going to take a half a teaspoon of baking soda and put it in to this one. And I'm going to stir it around. And notice what happens right away. It changes color to a bluish green. Okay? That's because baking soda is a base. And so it is a higher pH. And so it's changed its color to green as opposed to that purple. When it was purple, the pH was approximately 7. And so that's a neutral pH. But now we know it's a basic pH. Okay, at this point, we're going to take some clear soda, and we're going to add 1 tablespoon of the soda to that same glass. Now, a technique for chemistry is you never want to pour or measure over the container that you're putting something in, because if I pour too much and it spills over, I've just ruined my experiment. So I'm going to do this over this beaker to catch anything I spill in case I make a mistake. And so I'm going to add that and stir it. Now, why am I doing this? What I'm doing is I'm adding acid to this beaker. I had initially added base, which turned it purple. We're going to keep adding 1 tablespoon at a time of the acid until the pH gets lower and lower and becomes neutral again. And how will we know that? We'll know that by the indicator solution that turns purple. I don't want to add too much at a time because I want to make sure I can get it as close to that pH of 7 as possible. So I'm going to keep adding a tablespoon at a time and stirring. And you might notice it's getting a little more purpley blue now, which means the pH is going back down, becoming a little more acidic. Our goal is to get it back to that same color as the first one. We're getting closer. This takes patience, but of course it benefits us to do that. A little bit more. Now you'll notice my juice glass is getting full. If it ends up getting too full, I can just dump some of that back into the sink. The goal is not to have the right quantity, but to have the right components together. I'm hoping not to have to do that though. Let's see. We're getting closer. I'm going to move it a little bit so you can see better. Can you see how it's not quite there, but it's getting close to that purple color? Maybe 1 or 2 more tablespoons and we'll be there. 1. See if I can get down to level. Okay, so those colors, let me do one more. We're almost there. I want to get as close as possible. What I've done now is I now have 2 containers with a pH of 7. However, the container that I've added the baking soda and the soda to, I've got a buffer. And so what we're going to do now is we're going to slowly add vinegar to both of our containers and see what happens. Vinegar is a mild acid. Our indicator should turn a pink in the presence of a lower pH. So as I start adding a couple drops at a time to both containers and stirring those containers, we will see what a buffer does versus a solution that does not have a buffer. All right, so what I'm going to do, because here's another important thing, we never want to use or dispense material straight from our container. So I'm going to really quickly rinse out my beaker to remove any soda residue. Dry that off. So you're learning some good chemistry technique in our anatomy and physiology class, which is also a good thing. Rinse it with my distilled water. And now I'm going to pour some of the vinegar into my beaker so that I can dispense from the beaker without having to go in and out of my main container, because then I would contaminate potentially my main container. All right, I have an eyedropper, again, that I have cleaned ahead of time. And we're going to add 2 drops of vinegar into each of our containers. 1, 2. 1, 2. Ooh, I added 3, so I'm going to put 3 in this one. Okay, and I'm going to stir. Let me use my tongs to do that. Or actually, I have one for this container I'll use. And then I'll use the tongs for this one. So I don't want to cross-contaminate. Okay, so this is the container, remember, that does not have the buffer. This is the container that has the buffer. However, they were both at the same pH initially. Notice how they're different right now. Can you see how the one without the buffer has gotten a lower pH because our indicator solution is more pink? The one on the right has buffer in it, so it hasn't changed color. So note that in your lab notebook after, in this case I did 3 drops, but if you do 2 drops, do 2 drops at a time into both of them, and then make notation of that. And then I'm going to add 2 more drops to the container without the buffer, and I did 3 again, and we'll do 3. Always want to do the same number. I'll use this stir to stir this side. Let me again move it so you can see a little better. And my tongs to gently stir this one. Can you see what a buffer is doing? It is buffering it. It is taking care of that added acidity. And in the same way, the buffers in our body behave to keep our blood pH at that wonderful 7, neutral pH. So bicarbonate is a wonderful buffer that is one of the buffers in our blood system. Once you've done this, you can do a couple of times, a couple of iterations of continuing to add more vinegar into the solution that doesn't have the buffer as you add more vinegar to the one with the buffer until you see it change more and more. You eventually will begin to see this one turn very, very pink. This one's going to stay purple for quite a while. So continue to do that, log that in your notebook, and then clean everything up and put it away. Well, what did you see in the experiment? You should have noticed that the solution with the bicarbonate buffer didn't change color as the acidic vinegar was added to it, but the other one without the buffer did. That's because the bicarbonate buffer works to resist pH changes. So even though acid was added to its solution, the pH didn't change much because the buffer resisted that change. The other solution, the one without the buffer, changed pH pretty quickly, and that was noticeable due to the color change of the indicator solution. So you can see how buffers do work to prevent drastic pH change, and they do it rather quickly. Well, let's talk about exactly how it works to do this. A buffer is made up of a weak acid and a weak base. You made the buffer solution in the experiment with a weak acid from the soda, which is carbonic acid, and you used a weak base from the baking soda, which is bicarbonate. Now, vinegar, or acetic acid, is a weak acid. So as it was added to the container with the buffer in it, it was able to react with anything in that container. And being an acid, it most readily reacted with the base. In this case, it reacted with bicarbonate, or HCO3 ion. Remember that pH is a measure of the concentration of free hydrogen ions that are in a solution. More hydrogen ions, the lower the pH, or the more acidic a solution is. In this reaction, you see no free hydrogen ions as products, so this reaction doesn't affect the pH of the solution. Now, if there was no buffer in the solution, you would have a different result. The acetic acid or vinegar has nothing to react with, so it just breaks up or dissociates like this. And you can see that in this case, hydrogen ions do result in the products. This will lower the pH, making the solution more acidic, and that's just what happened in the glass without the buffer solution. Well, what would happen if we added hydroxyl ion, which is a strong base, to the buffer solution instead of acidic vinegar? That base would then want to react with the acid as you see in this equation. You can see that the hydrogen from the carbonic acid readily binds with the hydroxyl ion to make water. Thus, the addition of hydroxyl to a buffer doesn't alter the pH much. So to alter the pH of a solution, you have to increase or decrease the hydrogen ion concentration. A buffer slows that process down. Buffer solutions contain a weak acid and a weak base, so if acid is added, the weak base neutralizes it by chemically reacting with it so it won't lower the pH very much. And if a base is added to the buffer, the weak acid neutralizes it by chemically reacting with it so it won't raise the pH very much. It doesn't completely prevent pH change though. This is because even with a buffer solution, the pH will change a bit. It's because once, say, an acid is added to the buffer, the weak base within it is used up as it's neutralized that acid. So the concentration of weak base decreases, and that lowers the pH of the solution because there's now more weak acid in that buffer solution than the weak base. It doesn't change it much, but it does change it. We can say then that the buffer resists change in pH, but it cannot completely prevent change. If an acid is added to the buffer, it reduces the amount of weak base in the buffer, slightly lowering the pH. And if a base is added to the buffer, it reduces the amount of weak acid in the buffer, slightly raising the pH. Summing up then, we can say that buffers resist pH changes, but they cannot prevent pH changes. Of the three mechanisms of pH control, buffers are the least effective, yet they are still an important part due to their direct and immediate acid-base control. The body has three buffers that work to control acid-base balance. The first is the bicarbonate buffer system. In experiment 15.1, you actually made this buffer. It contains carbonic acid, H2CO3, and the bicarbonate ion, HCO3-. Bicarbonate buffer can be found in extracellular fluid. The carbonic acid component neutralizes any bases that get into the areas between the cells, and the bicarbonate component neutralizes any acids that are found there. The second buffer system is the phosphate buffer system. This buffer contains dihydrogen phosphate, H2PO4-, which is a weak acid, and monohydrogen phosphate, HPO4-2-, which is a weak base. This buffer system is found inside the cells because phosphate is commonly found there. It is also located in the nephron tubules. The third buffer system is the protein buffer system. It's located in the cells and in the blood plasma. Now, this system only contains proteins, so a single molecule acts as a buffer. You've already learned that the building blocks of proteins are amino acids. Now, the amino part of an amino acid is a weak base. The acid part of these molecules is a weak acid. So an amino acid is made up of both a weak acid and a weak base. Thus, proteins can behave as buffers. Remember that buffer systems are good in that they can react quickly to help control pH changes, but they are the least effective. It turns out that the second line of acid-base regulation is a bit slower, but it is more effective. It's the respiratory system. By controlling ventilation rate, it can help provide a longer-term control of the blood pH. Remember, as the ventilation changes in rate and depth, the body pH changes. If ventilation is increased, the carbon dioxide level of the blood will get lower because you are exhaling more of it, and that will increase pH. Decreased ventilation increases carbon dioxide levels of the blood, so it will lower pH. Do you remember how this works? When studying the respiratory system, you learned that varying rates and depths of ventilation change how much carbon dioxide is in the blood. And carbon dioxide chemically combines with water in the blood, forming carbonic acid. So the amount of carbon dioxide in the blood affects blood pH. The final line of defense to control acid-base levels in the body is through the kidneys. This is the slowest acting defense, however, it works the best. It's because the kidneys can directly remove hydrogen ions out of the blood or can keep them in the blood if needed. You already learned that this happens in the cells of the distal tubules of the nephrons. They're sensitive to pH. If they identify a decrease in pH of the interstitial fluid, which means that there are too many hydrogen ions there, as well as in the blood, they will increase the number of hydrogen ions that are secreted into the urine. By reducing hydrogen ion concentration of the blood, and thus the interstitial fluid, the pH will increase. This happens in the third step of urine formation, or in the secretion step, where materials are specifically picked out of the blood and put into the nephron. Hydrogen ions are removed from the blood this way. So if blood pH decreases or becomes more acidic, hydrogen ions need to be removed. So more secretion of hydrogen ions will happen in the nephron for removal in the urine. Additionally, if there's a decrease of blood pH, that means that the bicarbonate ion HCO3- reabsorption will have to happen in the nephron. That ion is a base, so if more base is reabsorbed into the blood, then the pH of the blood will increase. This then would decrease the pH of the urine because if bicarbonate is released into the blood, then there's less of it in the filtrate. So the filtrate and thus the urine pH would be more acidic. Now, if the distal tubules identify an increase of blood pH, the opposite thing happens. Fewer hydrogen ions are secreted by the nephron, leaving more hydrogen ions in the blood. Therefore, blood pH decreases. And the pH of the urine increases because there are less hydrogen ions in it. That increase of blood pH will also cause the nephron to reabsorb less bicarbonate ion, so less makes it back to the blood. That will lower blood pH, but the urine pH will increase because more bicarbonate will be there. So can you see the pattern here? If blood pH decreases, the nephron secretes hydrogen ions and increases reabsorption of bicarbonate. This raises the blood pH and lowers the urine pH. If the blood pH is too high, the nephron stops secreting hydrogen ions and decreases the amount of bicarbonate ion that is reabsorbed into the blood. This lowers the blood pH and raises urine pH.
Can you believe it took an entire module to cover the urinary system? At first glance, it seems to be a pretty small, insignificant system, but you now know that it is not the case. Just like this water treatment plant, your urinary system has the jobs of filtration, reabsorbing, and secretion, and each of these processes is a complicated process done by finely designed components. Just the design of the nephron with its impermeability and the ascending limb of the loop of Henle, plus its distal tubule and collecting ducts, ability to alter permeability based on ADH level. That's a system that is able to flex and change with the body's varying needs. And as I mentioned at the beginning of the module, the urinary system plays a vital role as it works with other systems of your body to regulate blood pressure, blood pH, and a whole host of other parameters. What a marvel of design!